eukaryotic and prokaryotic DNA, you can tell me, because you know that prokaryotic DNA is what shape?
Circular.
And our DNA is?
Linear.
How about exons and introns? Who has exons and introns?
Eukaryotes.
Do bacteria have exons and introns? So let's see if we can find that one up here. Here's the differences between eukaryotic transcription and translation and prokaryotic transcription. So eukaryotes, for instance, eukaryotic DNA contains introns. Remember, prokaryotic DNA does not. Here's some additional test questions. Did you know that transcription and translation does not occur simultaneously in eukaryotes? Where does transcription take place in eukaryotes? In the what? Nucleus. Where does translation take place? Outside of the nucleus, in the cell cytoplasm. Oh, that's pretty smart. Polycistronic, I want you to know what this means. Did you know that eukaryotic RNA encodes for only a single protein? But bacterial RNA encodes for many proteins at once. Therefore, bacterial mRNA is polycistronic. A strand of mRNA in a bacterium sometimes will code for 5 or 6 proteins all at once. That's called polycistronic. Here's another thing. Do you remember what the eukaryotic start codon was? What was it? Is that RNA or DNA?
RNA.
And if I were to ask you the DNA equivalent, it would be? By the way, does the eukaryotic start codon code for a amino acid? Yes. What's the name of that amino acid? Starts with an M. Methionine. Eukaryotic start codon is AUG, but it does not use formylmethionine. In other words, formylmethionine is the amino acid that bacterium use. We use methionine, bacterium use formylmethionine. So not only just these 4, I can think of another difference between the two, and that is eukaryotic DNA is linear and prokaryotic DNA is circular. But I might just ask you, what are the 4 differences between transcription and translation? Here are the 4 differences. That make sense?